Hello, my name is Victor Zberstis, and I'm on the team working behind the scenes on worldcommunitygrid.org, which has been harvesting spare processing time from computers around the world since 2004, putting those computers to good use while they would otherwise be consuming electrical power just waiting for that next keystroke or mouse movement. One of the research projects on World Community Grid is the Help Conquer Cancer project from the Ontario Cancer Institute in Toronto, Canada. They are helping researchers working to find cures for cancer by helping discover the physical structures of many large proteins involved in various forms of cancer. Please let me introduce Dr. Igor Jurisica, who will tell you more about the Help Conquer Cancer project running on worldcommunitygrid.org.
Thank you everybody up front for supporting World Community Grid and for donating the competing resources for this and other very worthy projects. What I will try to describe is not only the actual project that we run, but also the context in which we run it and why we do this kind of analysis. Currently, we are in the era where a lot of the biological and biomedical experiments are done by so-called high-throughput machines, by robotics. And this is both the challenge and the benefit, because now we have a lot more data available, but it's also a challenge because we have to manage this data and try to integrate it to understand what these individual pieces of the puzzle are telling us. So computationally, this leads us to developing multiple resources and also tools and algorithms for analysis and integration and interpretation of all of these different data. In our group, we focus on multiple aspects related to cancer. And one of the things is to understand better why certain people develop cancer, how we can improve their treatment or diagnose the disease earlier, so we hopefully will improve survival of the patients. So we are trying to identify prognostic and predictive signatures to help treat this disease better. Our other goal is also to understand biology behind it. So not only as a black box to help clinicians to triage patients into appropriate subgroups, but also to understand on a molecular, on a biological level, why certain things do happen or how can we potentially fix them or treat them. This brings us to the project that we do run on a World Community Grid, which is the Help Conquer Cancer, by running high throughput protein crystallography. We need to do this study in order to determine more structures for the proteins, and we need the structure of the protein to understand its function and potentially design new treatment options. Obviously, we cannot do this alone, and for that reason, we created a large consortium with the funding from Ontario government and also with many industrial partners, including IBM and World Community Grid, that helps us to integrate and analyze these diverse types of data for cancer, hopefully improving, as I mentioned earlier, survival of individual patients. Now, going into crystallography, which is one part of this large puzzle, the problem with crystallography is that it's de facto standard for determining three-dimensional structure of the protein. But as you can see on the numbers, we still have very small number of structures compared to all of the different proteins that we need to understand better. One of the reasons for this problem is also visible on the graph, that there are really sharp decline in terms of determining this structure based on the proteins that are available in different tissues in different organisms, including human. The problem for this is that there are many parameters that affect the crystallization, from purity of protein to many other different aspects. And we don't really understand how all of these work together. And that was one of the motivations for the study with our colleagues at the Huffman Woodward Medical Research Institute in Buffalo for speeding up this analysis. What you are looking at now is just one part of the lab in Buffalo that is focusing now on imaging the results from this large-scale screen. So the images that you see on your screen when the World Community Grid is running are produced by taking individual images from these small wells. There are 1,536 wells in each of these different plates, and that's one protein experiment. The scale really is enormous because the drop that you see on the right side is only 400 nanoliter large, and we try to understand, based on analyzing the image, what was the result of the experiment. So in principle, what we try to do is that we take these individual images and we try to characterize it by measuring multiple morphometric features. So we try to identify lines, gradations of shades of gray, and so on. And using combination of these features, although it's a large number, over 12,000, we build classifiers that can then identify if it's a crystal, garbage, or precipitate, or other results from the crystallization. Really, the challenges I mentioned is the numbers, because over the years we accumulated about 14,000 proteins that have been screened, and it's about 120 million images that currently are running on a grid. The importance of this is that this is going to affect not just cancer, although that's our focus. It's also going to affect other diseases, because over 1,000 labs are sending protein for these experiments. It's also good because it's a large variety, so we would be able to better understand the principles across a large scale of proteins. From a data viewpoint, by today's standards, is not that large. It's only about 30 terabytes. When we were starting, that was a challenge as well. But from the archival viewpoint, it's also challenging, because if you store one protein experiment on a one DVD, that's about 18-meter tall stack of DVDs, which is about 4 double-deckers on top of each other. So it's a massive amount of information that one has to tackle. The other aspect of trying to identify these results for the imaging is to ensure that what the computer is telling us is accurate. And so we compare it to the best experts in the field that classify the images, and we had a good training and testing set. The challenge is that there are multiple aspects that we have to consider. One is to design, in a sense, a clever algorithm that will extract important features from these images that would be useful for the classification. Obviously, considering the large number of these features, we also had to ensure that the analysis is optimized. So we optimized the compiler setup so we can run it relatively fast on a single CPU. But still, we need a high-performance computing platform of the size of World Community Grid to enable this backlog of data to be processed. Once we have the data, which is nearing completion, then we can analyze the results of these experiments as a first step. And in this specific view, we are looking into the classification of a three-way, only crystal, other, and clear, and 10-way classifier, which recognizes more features from these images. And the larger the red boxes, the better the performance, meaning that we identify all of the important components and also that we don't miss many. And the smaller the black boxes, the better, because those are the false positives and false negatives. And so as you can see, we are doing reasonably well, and importantly, we are doing comparably good to human experts. The next image is really just to highlight that the task is not trivial, because as you can go through any row on this set of images, you identify that true positives are quite similar to false positives and false negatives. So even humans do make errors in this analysis. But luckily, we don't need to be perfect for every single image, for every single protein. What we need to do is that in order to crystallize a protein, we need to identify conditions that are useful for optimizing the conditions for growing the crystal large enough so we can run it through a diffraction experiments and determine its structure. So the statistics from our analysis shows that looking just on the top 300 images out of the over 9,000 images will identify crystal if it's there. And importantly, 95% of these crystals will be triaged into the top 100 images. So importantly, we are likely not going to miss any crystals if they are there. Once we have this information, it's relatively straightforward to calculate what are really the benefits for the field, because we will roughly double the number of crystallizations that hopefully will lead to determining structures for important molecules. There are many optimization steps, and obviously even with a large screen that colleagues in Buffalo are running with 1536 conditions, we do not cover the whole space. But with the computational approaches from data mining, we can actually infer some of the missing data points based on the trajectories within the matrix, as shown in this specific diagram. Once all of the computation is done, even more analysis will be enabled. We have a relatively rich cube of data where on one side we have about 14,000 proteins and the corresponding information. On the other side we have 1536 descriptions of cocktails that lead to crystallization, and then we have six time points across the timeline over the month. And then each point in this cube is actually either a classification for the image, or it's actually 15,000 features that describe the morphometric characteristics of an individual image. So from this data, we will be able to identify which proteins are more amenable to what kind of conditions to determine crystallization conditions, which will lead us to identifying more possibilities for crystallizing a novel protein. One will be able to run it through the screen relatively easily and determine then similarities under which this protein likely will form a three-dimensional structure. So to conclude, what this analysis that through your donated computing power was enabled is to process the backlog of these 120 million or 115 million images. This kind of scale of analysis was never done in crystallography, and that's what we hope is that not only we will determine more crystal-bearing structures from already done experiments, but we will enable repository that will help us crystallize new proteins in the future. We will also be able to do this data mining and statistical analysis to determine principles that lead to crystallization, and that hopefully improve our understanding of how does it really work. I would like to really thank our collaborators at Hauptmann Woodward, George DeTita, Edward Snell, and Joe Laft. Obviously also IBM that helped us with optimizing code for running it on a grid. World Community Grid team, and definitely the users that donated the computing power. There are many funding agencies and other companies that help us achieve these goals, not only for protein crystallography, but in our effort to conquer cancer. Thank you very much.
We hope those listening are contributing and will encourage their friends to also contribute their spare computing power by joining worldcommunitygrid.org. We have time for a few questions. What does the three-dimensional structure of a protein do to help progress cancer research?
So as I mentioned earlier, in order to understand how the protein works, we need to understand its three-dimensional structure and understand other proteins that potentially bind to it. It's also, if we want to design new drug for a given protein that, for example, inhibits its function, we again need a three-dimensional structure of a protein. There are other methods except X-ray crystallography to determine the structure, but X-ray crystallography is de facto standard considering it can tackle proteins of different length, and most of the structures are still determined by this method.
What is the significance of a crystal being present? Can you briefly describe the X-ray diffraction process for finding the structures?
In order to determine the structure, one has to have a crystallized protein, because that's when it naturally forms the three-dimensional conformation. And X-ray diffraction experiment basically is after having a crystal, one shoots X-rays through the crystal, and depending on the diffraction pattern, there is a mathematical formula that enables to determine its structure. And we need to have a well-formed crystal in order to determine structure to a high resolution, so to understand details of the structure.
Would a low-gravity environment such as that found on the International Space Station help optimize conditions for protein crystallization? Would this benefit your research?
There are definitely, or there have been definitely efforts to do some crystallization experiments in a zero gravity. As I mentioned earlier, one of the issues is that crystallography or crystallization process is dependent on many different factors. Gravity might be one of them, but then there would be other aspects to optimize. So the effort would really require to replicate, in a sense, this high throughput screen in a zero gravity setting and identify which proteins might be more amenable to the crystallization in that form.
Can the Human Proteome Folding Project do the same thing?
As I mentioned earlier, there are basically three main methods for determining 3D structure of a protein. It's X-ray crystallography, NMR, and then protein folding. And they are to some degree orthogonal, but they can benefit from each other as well. At the present time, for high resolution determination of the 3D structure, really NMR or X-ray crystallography would be the choice. However, the data that we are generating and the structures that will come out of it will help improve algorithms for the protein folding projects. And also, in some cases, knowing some at least rough idea of the 3D structure from a protein folding may help us to identify if we can crystallize the protein alone or we need some binding partner for it. And thus, also the protein folding can help with the crystallography.
Can you tell us more about using GPUs, graphics processing units?
We started with the CPU as the first version, but then as the GPU platform became more available, we started to look into the possibility of running it there. One of the reasons also was that while World Community Grid enabled us to process this backlog of data, eventually moving into the lab-based setting, we would need to have a relatively powerful machine that would help us to analyze these images in real time or close to real time, but at a fraction of the size or cost of World Community Grid. And so GPUs were one of the platforms that we looked into. And kind of our preliminary analysis shows that obviously there is a variance in terms of performance depending on the type of a graphical card, as you can see on the right side of on the bar chart. But importantly, we can shrink the computation from about an hour on a CPU to about a minute or two on a GPU. So it significantly reduces complexity. And one of the reasons is that we do basically image analysis, which GPUs have been designed for, and so we can nicely optimize it. So we are also looking into nice comparative statistics of how the GPU run on a World Community Grid will speed up finishing of this project.
I might add that we've been beta testing it, and we expect to go into production with that soon. Exactly what are graphics processing units?
So it's a graphical processing unit, and it's basically processing units that are on a higher end graphical, available on a higher end graphical cards. Usually they run in hundreds, and thus on a high end graphical cards, one might have available 200 to 300 processing units, which compares to couple of cores available on CPUs, at least at the present time. So for some of the matrix calculations, this is a very useful platform.
How many more PCs would be helpful to help accelerate the project?
Based on the statistics, it seems that we are getting about 54 CPU years a day since the inception of the project in 2007. And any new computing device obviously speeds up how many more images we can process in a unit of time. But I didn't really calculate how many more would be needed for us to finish by, say, Thanksgiving. I don't know.
What are the most important factors that affect protein crystallization?
As I mentioned on the second or third slide, actually fourth, there are multiple factors. There are basically purity of protein is one of the aspects. So for many proteins, especially membrane proteins, it might be difficult to get them in sufficient quantities for the crystallization experiments. There are other aspects that different proteins will have a different requirement in terms of the temperature or pH that will determine if they will be amenable to crystallization under the given conditions. At the present time, the standard is that people try existing screens to identify reasonable conditions and then try to optimize them. The problem with many proteins is that you run it through the screen and there is nothing, no result, no crystallization result. So there is no easy step for the optimization. So people try other screens and keep trying and keep trying. And so that's one of the reasons why some of the, for example, membrane proteins are very rare or difficult to crystallize.
How many protein structures have been found with the help of World Community Grid data so far?
We have been estimating this number based on maybe let me backtrack. So there have been about 14,000 proteins that have been screened since the start of this high throughput facility in Buffalo. And we don't have result about every protein that has been sent through the screen of what happened with it. But based on a large number of experiments that we have tracked on, we are estimating that it's over 3,000 structures that have been determined just from those screens.
Does your project do anything with the protein structures that you've already determined, or who do you pass them on to?
As I mentioned, these proteins have been sent by biologists from over 1,000 different labs across the world, and many of them are related to cancer, many of them are related to other diseases or basic biological functions, and it would be those biologists who would be following up with optimizing and studying the function of these proteins. In our own work, we take the results from kind of aggregated results, so not only from this screen, but also from other efforts or single labs from Protein Data Bank, where there are now about 83,000 structures, almost 84,000 structures. And we use that integrated with other information about, for example, networks of physical interactions among the proteins, or our new resources that store information about drug targets, structure of these drugs, and how potentially they can bind to these proteins or how they can affect the function of these proteins.
Thanks again, Dr. Jurisica, and those attending. Sorry if we didn't get to your question. Please use our forums at worldcommunitygrid.org to post any additional questions. This concludes our webcast.
Thank you again.